The goal of this video is to discuss the anatomy of the neuron. Certainly, the main focus of this course is human physiology and the physiology of neurons, not specifically physiology of neurons, but we'll talk about the physiology of neurons in depth. But to do that, we really need to get a good understanding of what a neuron is and all the different components of a neuron. So first and foremost, a neuron is just a nerve cell that is part of the nervous system, and we'll talk about that in more detail. Generally, I draw cells as a very symmetric circle, which is totally unrealistic. And when we look at the neuron, that'll be a prime example of why we shouldn't always think of neurons, as cells as circles, because they are highly variable in their shape and their morphology. It's Corona Summer 2020, and it's also a time where our country is rife with angst, protest, arrest. I just want to take the time to suggest one thing, that to a certain degree, I think we should all be stoked that Donald Trump was elected president. And there's two ways to look at this. If you're a Trump supporter, if you're from the right, which I respect you because everyone's entitled to their own opinion and to vote any way they choose. But if you are a supporter of Donald Trump, you should be stoked because he got elected and he's promoting policies that he outlined and theoretically that you voted for. But if you think Donald Trump is a clown, you should also be stoked to a certain degree. That's not to say you should be stoked how he treats people, his approach to issues in our country and globally. But it is the system that elected Donald Trump, an outsider, granted an outsider with a lot of money, but there have been outsiders who have run for president with a ton of money before that did not get elected. So he's a complete outsider, if not an outlier in his personality and his approach to dealing with people, policy issues in this country. But most importantly, the way I look at it, this is the same system that allowed us to elect a black man for not one term, but for 2 terms to lead this country for 8 years. And it may be hard to really grasp the significance of that, especially considering the challenges facing our country right now. I don't know who ever really thought that it was possible that a black man would be elected president of the United States of America. But due to our system of election of presidents, and truth be known, I'm not a expert on political science or government, but we were able to elect a black man for president of the United States, and that is huge. But it was that same opportunity that was given Donald Trump, and he was elected president, whether you like it or not. So keep that in mind. There's going to be another opportunity, other elections down the road. We have to do our part to agenda that would be best suited for our country. And that's all I have to say about that. Okay, so back to anatomy of a neuron. We'll take a look at the anatomy right here. And once again, the neuron looks different than most cells we've talked about. That big circle right here is the cell body. And that cell body contains the nucleus and a great majority of the organelles of the neuron, which once again is a nerve cell. So this big circular region right here is the cell body. Leaving the cell body from here all the way down here, the elongated structure is known as the axon. So from here down to here is the axon. At the far right, all of these are axon terminals. And the axon terminals contain secretory vesicles, which themselves contain signaling molecules known as neurotransmitters that will be released when stimulated. And we'll talk about that. So we have axon terminals, axon, cell body. On the far left here, all these appendages or surface modifications are known as dendrites. And those dendrites serve only one purpose, and that is to increase the surface area of the cell body. Peppered along all of these dendrites are going to be ligand-gated ion channels. And by having the dendrites and increasing the surface area, we can exponentially increase the number of ion channels that are embedded within the dendrites and within the cell body. If we didn't have the dendrites, there'd be much less surface area to the cell body and much less ion channels and less opportunity to excite this cell. And excite really is the operative word because neurons are excitable cells, extremely excitable cells. And when they become excited, they release their signaling molecule known as neurotransmitters. So if we look at this other picture of a neuron right here, the only difference that I'm showing here is a blown up view of the secretory vesicle, which contains, and secretory vesicles in neurons are also known as synaptic vesicles, and we'll cover that. So I have been using the term secretory vesicle. Generally with a neuron, we do refer to them as synaptic vesicles, though truthfully they're the same thing because they contain signaling molecules in them. But when those signaling molecules get released, they induce some sort of response or stimuli downstream of this neuron. So what causes them to be released? Well, that's going to be the topic of a subsequent video. But in short, it's going to be an action potential along this axon, which stimulates the exocytosis of these neurotransmitters. All of that is going to be initiated down here at the dendritic end of the neuron, where we're going to have a massive influx of sodium, which initiates the depolarization event, which travels all the way down here to release the neurotransmitters. That's it for your neuron anatomy. We'll get more in depth on the physiology of that in the next video.